Now I want to introduce Professor Pollack from Seattle, Gerry Pollack, who's become a very good friend over the last 9 months. We've had emails going all kinds of days and nights. But actually, he received his PhD in biomedical engineering from the University of Pennsylvania in 1968. He's the founding editor of the journal Water, convener of the annual Conference in Physics, Chemistry and Biology of Water, and executive director of the Institute for Venture Science. Along the way, he's been presented with 2 TEDx awards on water. In 2015, he won the Brand Glorious Award, previously bestowed on notables such as Nelson Mandela, Hillary Clinton, and Steve Jobs. And in 2016, he was awarded the first Emoto Peace Prize. Professor Pollack.
Thank you. Yeah. Well, thank you, Aaron, for the invitation, and thank you all for coming to this really special event. So I want to talk about the 4th phase of water, and particularly in general about water. Now, I think for many people, it must be that everything that there is to be known about water is already known, because it's such a simple molecule. It's been studied for eons, and surely by this time we must know everything that there is to know. I'm going to open with a few slides which I hope will demonstrate that there's a lot about water that we don't know at all. These are simple slides, and so we start with a very familiar scene, one cloud over a body of water. If you think about it for a moment, though, how is it possible that there's only one cloud above, because the water is evaporating from everywhere? There should be one continuous cloud at all times. Sometimes there is, if you live in Seattle. But more ordinarily, you see a cloud like this. And so what's the explanation for this? How do you get to it? How do you explain it? And also, I'm not going to talk about this, but what keeps the cloud floating in the sky? If you think about it, the cloud is water, little water droplets. And water is heavier than air. You expect that gravitation would be pulling the cloud down, but sometimes it does, but sometimes it doesn't. And what's the explanation for that? So this is water, and we really don't understand it. Here's another example. This is water droplets being released onto a surface of water, and you can see what happens. The two should coalesce instantly, but that doesn't always happen. This comes from our laboratory, and other people have reproduced this. It's not strange. So what's the explanation? You don't know? Okay, well, I'm not sure either, but okay, here's from Elmar Fuchs. 2 beakers of water, and the lips are touching one another. And you take 2 electrodes and plunge them into the water, high voltage between them, and here's what happens when you turn on the voltage. You get a bridge between the 2, and if you pull one beaker away from the other, see, you get a bridge that persists, and this bridge can persist for essentially indefinitely over a distance of up to 3 or 4 centimeters. So what's the explanation? It's not so obvious what the explanation is. It's water. You don't expect water to behave that way, but indeed it does. Now here's a 4th and final demonstration. So what you see here, that blue is a magnet. It's a superconducting magnet, and it's sitting just behind a trough filled with water. And the water is colored red for effect. And you turn on the superconducting magnet, and the water splits so that Moses could walk across the Red Sea. Demonstrated in a laboratory. You've seen it. Okay. Well, my interest started with a guy named Gilbert Ling. Gilbert is now, if you do the arithmetic, he's 99 years old, alive and kicking. And by the time I met him, he'd already written 5 books, and I think 2 more, maybe 3 more since then. Gilbert was a scholar who came from China, the first group of scholars to come after World War II. And one of them, Yang, won a Nobel Prize in physics, and they all thought that Gilbert was the cleverest of all of them. And Gilbert was controversial. Anybody who puts forward something unusual is controversial. And Gilbert provided evidence that the water inside the cell was not like the water sitting in a glass, but that the molecules of water were organized in some way. And I became seriously interested in this. We were studying muscles, and when people talk about muscles and muscle contraction, they talk about the proteins inside the muscle that give rise to contraction. But of course, muscle is mostly water, and not many people are thinking about the water that's inside. And so I began thinking about this and thinking about Gilbert's monumental work. And I decided that because Gilbert's work is sometimes he's a physical chemist, and his work can be understood by some physical chemists. It's not so obvious. And I decided to write a book, a kind of popular book, to popularize Gilbert Ling's ideas. And the book is called Cells, Gels, and the Engines of Life, and it came out in 2001. And it's really obvious how popular it is. Well, out of Gilbert's work in this book, I transitioned to becoming really interested in water, and especially in ordering of water, ordered water. And the idea is sort of summarized here from Gilbert's ideas and presented in my book, is that inside the cell you have proteins, and the proteins usually have charged surfaces that are exposed to the water. And the water is shown here as little dipole, plus, minus, and you can get the idea that Gilbert was trying to put forth here, that next to a charge on the surface, a water molecule would stick. So if you have plus here, you have minus here and plus here, and so on. And therefore, you get molecules lining up. Now, the establishment point of view is that this kind of ordering may exist for 1 or 2 molecular layers, but not more, because the effects of thermal or Brownian motion tend to give rise to promote disorder. So by the time you have 2 or 3 layers, you've got no more order. Gilbert argued that's not true, that you can get ordering that extends out maybe to dozens of molecular layers or more. And we thought this is really interesting, and of course we wanted to pursue experimentally. Having written the book, nice to do some experiments to try to figure out what's really going on, get some details. And we started doing that based on this following idea or principle, that when you have order, as in a crystal, this would be a kind of liquid crystal, that the ordering would tend to exclude solutes and particles. It's kind of like when ice forms. In order to get a perfect crystal, it has to push out all the impurities, and so we were trying to identify a possible experimental model where there would be a region where solutes and particles were excluded. And through some quirk, we found one, and this is one of the first slides. So into a chamber, which would cover the entire slide, you have a gel, and we put a gel there, and we added water and particles. And these particles were little spheres. They're called microspheres, and they're used widely by many scientists. You can see these little ones here. And we noticed very quickly that there was a zone where there were no particles. And this is a pretty big zone. Right here, you can see 10 micrometers. That may seem awfully small, but by molecular standards, it's pretty large. It turned out to be even more dramatic than that. If you watch the video, this is one of the first ones we took, you can see that it looks like something is growing and pushing the microspheres all the way out. And then the video stops. And this zone here is about 50 micrometers, so it's a pretty huge zone. And we had the idea, maybe, possibly, this is a zone where the water molecules were ordered. We didn't have any evidence at the time, but it seemed to fit the general idea. Well, we began seeing this again and again and again, and a colleague from Australia said, you know, you ought to give it a name. And so he suggested the name exclusion zone because of obvious reasons. It excludes. And he said, well, one of the advantages of exclusion zone is EZ. It's easy to remember. So it doesn't work all over because some people use Z instead of Z. So, but easy. Anyway, another example here is of the exclusion zone is a piece of naphthalene. Naphthalene as a polymer is a bit like Teflon, except that it has charge groups associated. And it comes in a nice, convenient, parenthesis, expensive sheet. And you take the sheet and you can cut it in any shape you like. So you can see we cut it in an arrowhead shape. And you put it down, plunk it down to the bottom of the chamber, and you do the same experiment. You have microspheres suspended in water, and then you have an exclusion zone that sits here. And again, you watch it as a function of time, and you see it grows. And in this case, it grows to 400 or 500 micrometers, very large, almost half a millimeter. And it does it reliably and consistently. And so we began using naphthalene quite a lot for many of the experiments because it's just so easy to use. You just take a sheet and cut it. Well, since we identified this, a lot of people have tried to duplicate it. And this is a partial list of people. This was put together about 6 or 7 years ago, and many people have actually done it. In fact, to my chagrin, it was published, the same experiment was published in 1970 by a Japanese group who were looking at the lens of the eye, both the natural lens of the eye and artificial lenses. And they did the same experiment. They put it in microsphere suspension, and they noticed a zone right next to it, roughly the same size as we found. So anyway, what we found is old hat. It's nothing new. So what I want to do is get to the basis of all this, and I want to answer 5 questions. The first question, is this exclusion phenomenon general or just a couple slides that I showed you? I think you know the answer. Does it really arise from the ordering of water molecules, or is there something else? And I think you know the answer already. Can water ordering explain those first few slides that I showed, whose interpretation you couldn't figure out? At least I think so. And in order to create order, you need energy. Otherwise, we tend toward disorder. And so where on earth does this energy come from? Not obvious at all, but the answer turns out to be rather simple. And might these findings apply broadly? And in terms of the second question, does it rise from water ordering? I want to, as I go through this, I want to talk about water memory, because in order to have memory, you need something that's ordered and stable. And we found something that's ordered and stable, which could be a substrate for information. I want to tell you about that as I go to question number 2. Okay, first question, generality. So in terms of generality, the first question is, well, what kinds of surfaces produce this kind of order? And I don't want to take you through a big list, but we've looked at at least a dozen, perhaps closer to 24 different gels, various polymers. We looked at biological surfaces of various sorts, and we even looked at monolayers, that is single molecular layers on gold. It's a standard preparation, and we did not find it on every one, but on most of them. And so it's fairly general. The monolayer experiment is interesting because it suggests that if you can get this EZ next to a single molecular layer, that the single molecular layer may act as a template, which then organizes what's beyond. And this this is a common phenomenon in dealing with semiconductors. You get ordering that goes way out without any transfer of material. So anyway, the point is it's pretty general. In terms of the solutes are excluded, that's the second part of the question, which solutes are excluded and which ones are not. So we tried many substances, either suspended or dissolved in the water, starting from cells, for example, bacteria. They're out, large particles, small particles, big molecules like proteins and so excluded, down to rather small molecules. And when you get small enough, it becomes a real challenge to test to see whether they're excluded or not excluded. But here's an example of a dye which consists of molecules, a mixture of molecules that are roughly in the order of 100 Daltons molecular weight. And that's excluded. And I'll show you. And this is what we use as a dye. And some of you know, if you remember your high school chemistry, litmus paper, you stick, changes color with pH, one color, low pH, high pH. So the chemicals that are put into the paper, you can actually get dissolved in water. So it's a liquid, and it does exactly the same thing. So we put this liquid, we took a piece of Nafion at the bottom of the chamber, and you have an exclusion zone here and water here, and we put this dye in the water to see whether the dye is excluded. And the next slide shows a result. So you can see here at the bottom is a piece of Nafion, and this is water, basically, which contains dye. And the first thing you see is, oh, this is a pretty interesting distribution of colors. You might even want to frame it and put it, you may not, but yeah. At any rate, what I want to illustrate in this particular slide is that there's no dye in the exclusion zone. The EZ is sitting right here, and it's clear. I don't know how well you can see it with the lighting, but I can see it on my screen. And the second is of course, the effect here. And the colors correspond to particular pH values. And so the pH here is very low. It's pH 3 or less in order to get this red color. So you have a lot of protons here and fewer, and then you're back to neutrality here. So there's a gradient of protons. And these protons, this positively charged species, are very important, and I'll come back to it in a couple of slides. So, but for now, the most important thing is that these dyes, mixture of 5 or 6 chemicals, molecular weight, roughly 100 or so, are excluded from the exclusion zone. So in terms of generality, we can say that many charged or hydrophilic water-loving surfaces generate exclusion zones. If you remember, hydrophilic means water-loving, and if you have a surface that is hydrophilic and you put a drop of water on it, the drop spreads out, as opposed to, for example, Teflon, hydrophobic, fearing water, where the surface doesn't like the water, or the water doesn't like the surface, and it beads up. It has almost no interaction with the surface. So this is very common among hydrophilic surfaces. Question 2 is this zone really different from bulk water? And I've alluded to the possibility that it might be, that there's ordering that goes on there, but I presented no evidence. And I'm going to list the evidence. I'm going to list eight pieces of evidence. There's more. By now there are 11 or 12, but I don't have time. I think you'll get the idea. I'm not going to go over each one in detail because it's been published and you'll be bored to death. I would just want to talk about one of them, so I'll get to it. The first is that easy water molecule. Water is very useful. Easy water molecules are more constrained than ordinary water molecules, and we used NMR, nuclear magnetic resonance, to determine that. The second one is that the easy molecules are more stable than ordinary water, and we used infrared radiation coming from the specimen to identify that. And the third, this is a kind of surprising one, is that EZ typically has negative charge. It's not neutral. Water is neutral. This is not neutral. And I'll show you those measurements afterward because they're central to what I want to talk about. And EZ absorbs light in the ultraviolet at 270 nanometers, and ordinary water doesn't do that. So again, these are differences between EZ and ordinary water. EZ is more viscous. It has a consistency of like honey or raw egg white. The molecules inside the EZ are aligned with one another in some way, as Gilbert Ling had suggested. The molecular structure is different from ordinary water. And finally, the optical properties are different. Two Russian groups published in the same year. They don't know one another, and they got the same result, that the refractive index, the bending capacity of light, is up to 11%, same number from both groups, higher than H2O, than ordinary water. One used a biological sample and the other a physical sample, but the results were pretty much the same. So I just want to go over one of these, the EZ as negative charge, because this is really important. And this experimental setup is shown here. I apologize for the complication. So imagine a chamber here, and where it says inside, it means the inside of a gel. So this is a gel. It's a polyacrylic acid gel that we put here, and the outside is water. And so if you remember, at the boundary between the gel and the water is an exclusion zone, which for the polyacrylic acid gel is roughly a couple of hundred micrometers. And we wanted to know whether the electrical potential in here might be different from ordinary water. We didn't know. We didn't even think of. And I remember I was with a Russian colleague, friend of mine, and we did the experiment together. And the experiment is taking two electrodes. If you want to measure a potential difference, you need two electrodes. So we put one electrode somewhere out here is a reference electrode, a microelectrode, incidentally invented by the same Gilbert Ling, whose picture I showed. So one is out here, and the other one is a movable electrode to be moved with a motor that went from position to position. So when we place the electrode here, which is pretty far from the interface, then the potential difference between this point and the reference is zero. It's what we expected. You put two electrodes in water, you get zero potential difference. Comforting. So then we moved the electrode closer and closer. And once we got in the region of the exclusion zone, we began to pick up a negative electrical potential. The closer to the boundary we got, the more negative the potential. So it looked as though the exclusion zone, at least for the polyacrylic acid gel, had a negative electrical potential. Now, it surprised us, and I remember that my friend Felix was surprised as well, and his wife, back in Russia, was doing pretty much the same kinds of experiments, experimenting with gels and microelectrodes. So he called, and he said, honey, or whatever the equivalent is in Russia, in Russian, would you please try the experiment? Within a few days, we were confirmed that the same phenomenon occurs in Russia as in Seattle. So we were happy about that. Then we thought, well, maybe there's something peculiar about this particular gel. So got rid of the gel, and instead of the gel, we used a piece of Nafion. I showed you about Nafion. It gives rise to a nice, sizable exclusion zone. And we did the same experiment, and you can see the result. It's not, again, the exclusion zone is negatively charged. The correspondence is not perfect, but essentially the same result, that it's a negative electrical potential. So at any rate, it looked to us as though the exclusion zones were not neutral, as we expect, but negatively charged. We scratched our heads because this doesn't make any sense at all. Imagine the experiment. You have a chamber, and you have a gel, and you pour in some water, and this water is neutral. So how is it possible that you take neutral water and you put it in, and you get a giant zone by molecular standards, a giant zone that's negatively charged? It's like magic, it doesn't. And we thought, well, the only conceivable way that this could happen is somehow if the water molecule itself was split into the negative and positive components, H plus, OH minus. So if that's true, then you'd expect if this zone were negative, somewhere else there had to be some positive charge, for example, somewhere out here. And if so, it would be an indication that whatever we were doing is somehow creating a splitting of water. And is there any evidence for a positive charge somewhere else? Well, the answer is you've already seen the evidence. Remember this colorful slide? So it's now turned 90 degrees. And this exclusion zone that you see here contains a negative charge as we measured with the microelectrodes. And remember, the pH in this zone is 3 or less. So this has a huge concentration of positively charged protons. So it looked as though there really is a separation between negative charge and positive charge, implying that the water molecules are splitting. But just to be sure that we weren't fooling ourselves, it's easy. We all love our own hypotheses. So we put 2 electrodes, one electrode here, and connected them through a resistor. And if there really is a separation of positive and negative charges, then we expect current flow from the positive to the negative. And this is one of the first records showing, indeed, that we get current flow. This is current versus time. And you can see it starts at some, quote, high level, goes down to some baseline. It's not 0. Here's 0. And it's pretty much maintained for a substantial period of time, which confirms that there really is a separation of charge, because if there were no separation, we wouldn't get current flow. So we have a separation of charge, which basically means that we have essentially a charged battery in water. It looks something like that, where this is the exclusion zone, and the ordinary water beyond has the opposite charge of the exclusion zone. So going back to the previous slide, we looked at this, and indeed, typically, the EZ has a negative charge. Okay, now what about the structure of this exclusion zone? If you look at these 3, the blue ones starting here, the molecules are aligned, and they're relatively stable and constrained. This has the characteristics of a liquid crystal, as I mentioned before. So far, let me summarize where we are. We have water sitting next to a hydrophilic surface, and the water is liquid crystalline of some sort. It's got negative charge, not neutral. It excludes solutes profoundly, and there's some indication that the correct structure is not a stack of dipoles, as Gilbert Ling has suggested, and as indeed we suggested too in that 2001 book that the baby preferred. There's something, and I'll go back to that in a moment, because in order to understand the function of this zone, you need to know the structure of the zone. And also, it may extend very far from the so-called nucleating surface where this begins to grow. Now, how far is very far? Well, I told you that the standard view is 1 or 2 molecular layers. Gilbert Ling suggested dozens. This, if correct, is millions, several million molecular layers, if the evidence is correct. It was suggested very long ago, more than 100 years ago, by Sir William Hardy, famous British scientist, colloid chemist. He said something is the matter, because even at that time, it was known that there were many anomalous features of water that couldn't be explained by the ordinary 3 phases, the solid, liquid, and vapor. He suggested that there must be a 4th phase of water, and in order to explain these various anomalies that simply couldn't be explained with the standard paradigm. And so it's possible that what we've discovered is what was suggested by Sir William Hardy long ago, that there's a 4th phase of water. Now, I suggested to you that the dipoles might not be correct. Why why did I suggest that? Well, it's a sort of obvious problem when you think about it. So dipoles are neutral. Dipole has a plus and a minus, and they're equally charged. So dipoles are neutral. And we measure that the EZ has negative charge. And so this is a problem, because you can take dipoles and you can stack them, you know, from here to the moon, and you'll never get a negative charge. But we measured a negative charge, and therefore dipoles can't be correct. There's something basically wrong. There's another problem, and this, I mentioned that the EZ absorbs light in the UV at 270 nanometers, and this is usually characteristic of ring-like structures with delocalized electrons, not dipoles. And so those are the two reasons. Now, so where do you go from there? How do you deduce what the correct structure might be? And what we thought is that the structure of the EZ should have some precedent. In other words, in all likelihood, the structure is going to be similar to another known structure of water. Not the same, but similar. So what structure of water do we know about? Well, certainly not vapor. We don't know about liquid water, surprisingly. There are many theories of the structure of this kind of water, and the only similarity among them is that the person putting forth the idea suggests that everybody else is wrong. Okay, so we a touch of arrogance there, but we don't know. But ice is different, because ice is a crystalline structure, and when something is ordered and crystalline, you can use diffraction to deduce what the structure is. And so we kind of started with this principle, and we said, let's take a look at ice. We know the structure of ice, and maybe the EZ structure is some variant on the structure of ice, better than pulling some arbitrary thing out of a hat. What's the structure of ice? Well, it looks something like this, and I promise you that ice is not red. But so the red ones here are oxygens, and the first thing you see is the characteristic honeycomb or hexagonal structure, and the second thing is that it occurs in sheets which are stacked on one another. And you'll also notice, those of you who are still awake, that that it's not that ice has not only oxygen but hydrogens. So where are the hydrogens? Well, they're just omitted for clarity, but they would appear halfway between each pair of oxygen. So there's one here, 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 and so on. Okay, so this is the structure. Now, if you look at this structure from a different angle, same structure, different angle, then you can see that ice is not only red but blue. So these blue dots here are protons, and the idea is that you have a positive proton cross-linking these negatively charged oxygens, and so basically what that does is it glues together these oxygens, and that's why ice is solid. So this is the structure of ice. So we had the idea, how can we modify this structure in such a way that it might conform to the properties of EZ that we would be expecting? And you know, the obvious is to remove the protons. If we remove these blue dots here, then this structure goes from neutral to negatively charged, because we're pulling out positive charges. And that's what we want. We want negative charge. And also, since we're removing the glue that keeps these together, glued together, it's not a solid anymore. It's not rigid. And so we have a structure that, and the EZ is not rigid. It's kind of gel-like. And so for a good while, I thought we had it. I thought this made a lot of sense based on precedent. Until a colleague tapped me on the shoulder and said, well, good try, friend, but there's a minor problem, and that is it's impossible. Thank you very much. Why is it impossible? Well, it's impossible because if you pull out the positive charge, then you have two negative charges that repel one another, and because these two oxygens and others, of course, repel each other, the whole structure will fly apart. So minor problem, it's an impossible structure. So after a few weeks of antidepressants, I really thought I thought we had something, but this criticism was pretty clear, and it made a lot of sense. I remember on a flight back from Japan to Seattle, right halfway through, thinking, I got it. And I think this structure that I'll show you in a moment has real promise. And so that is, you just take the two layers and shift them by a small amount, half of the oxygen-oxygen distance. So you see two layers here. The one closer to you has been shifted to the right by half of the oxygen-oxygen distance. And then you get something really nice, because then what you have is that you can see that the negative oxygen from the plane behind is aligned with the positive hydrogen in the plane closer to you, and they stick electrostatically. So you have plus and minus right next to each other. The same thing occurs here, and wherever you see a dashed circle, you have stickiness. So the planes now don't fly apart. They stick to one another, and they stick electrostatically. It's not a strong bond, it's a weak bond. And if you were to shear it enough, it would come loose. Think about egg white. Think about if you shear the egg white, raw egg white, just a little bit, it bounces back. But if you shear it enough, then it keeps flowing. And this structure would have exactly that characteristic. So we think we're on to something, and the way it would work is you have some hydrophilic material, the nucleating surface, next to water, and out of the water grow these layers, these EZ layers, one after another, and you can see it basically just keeps building up from the water. And if you look at more detail at the surface of one of these planes, it looks something like this. It's hexagonal, just like the ice. And if you count the number of oxygens and hydrogens in one unit cell, it turns out that it's not H2O anymore, it's actually H3O2. And so if you think about it, at first it's sort of jarring, and how could that be? But if you think about it, it can't be H2O because H2O is neutral, and this plane must be negatively charged to conform to the experimental results. So we think we have H3O2. If you had H4O2, it'd be neutral, but now you see it's negatively charged. Now, in order to achieve what we achieved, if you remember, what we did is we shifted one plane to the right by half of the oxygen-oxygen distance. And what happened is that, remember, the negative charge lined up with the positive, and the positive aligned with the negative. So you have stickiness at those points. But I didn't have to shift it to the right. I could have shifted it to the left, or I could have shifted it 60 degrees or 120 degrees and gotten the same result because of symmetry, right? So why is that important or useful? It's useful because with this flexibility to move this way, this way, this way, you can stack these to build interesting structures, and one of them is a helix. So here you can see a helix. This is the primary layer 0. Layer 1 is shifted to the right by 0 degrees, 60 degrees, 120 degrees, and so you get a nice helix. So why is the helix necessary? Why is it valuable? It's valuable because in biology, so many things are helical, right? So you'll hear about DNA from Professor Montagnier and RNA and helically oriented or helically wound proteins, of which there are many. And so it's known that next to these structures is some kind of structured or ordered water. And the model that I've demonstrated to you can conform very nicely because it can adopt this helical motif to fit very nicely next to a DNA, for example. So the advantages, so to speak, advantages of this non-dipolar EZ is, number one, precedent. The structure is not ice, but it's not so different from ice. And I'll come back to that later because it's important. It has negative charge as the experimental results dictate. The ring-like structures can perhaps explain the characteristic 270 nanometer absorption of the EZ. It's able to accommodate helical structures. And finally, the crystal-like structure it turns out that this can be solidified. So, and to give you solid water at room temperature. And this is not done by us. This is done by Vittorio Elia and his colleagues in Naples. So here, this looks like a cloud, and indeed it does. To get this, here's what they did. They put a sheet of Nafion into a chamber. They let the EZ build up, and then they rolled over it to collect the EZ and some water. And they poured more water, let it build up, and they did the same. And they repeated this hundreds of times. And they put it into a beaker. And typically, you know, if you put water into a beaker, let it sit for a while, it looks pretty much like this. But theirs looked like this. And so they deduced that this must be a region that is like EZs. But then what they did was they put it into a lyophilizer that protein biochemists use to get rid of water. So you have a kind of dried protein, but the dried protein still has always has some water attached to it. It's hard to get rid of all the water. And anyway, when it came out of this lyophilizer machine, then they obtained, at the bottom of the flask, they obtained essentially EZ water at room temperature. And we got some of this from Vittorio. It feels like, it looks like a kind of wound thread, and it's solid, and if you put it into your hands, it kind of comes apart pretty quickly. And they did mass spectrometry on this, and you know, many people say, wow, it must be some kind of contaminant or something, but they found essentially no contaminants. So this is remarkable because this is solid EZ water at room temperature. Imagine solid water at room temperature. So if you think about memory, so you know, we all think about memory and such, and this contains atoms of silicon or silicon dioxide, and you all know that these each one of these can either be equivalent of a 0 or a 1, different locations, and this is proved remarkable in benefits to humanity and also some problems. But if you think about the EZ, it actually has a kind of similar character. And remember, it can be solidified at room temperature. So think about this model. We have the nucleating surface here, and we have layers. And this is basically semi-solid here. And the hypothesis is that just like in the computer memory, you can store information in this array of atoms that exist here. So usually each surface of each molecule is distinguished by the array of charges on the surface. So we might imagine a generic surface with no real information, that is, it's got plus, minus, and so on in this very regular array. And next to the surface, it nucleates some EZ layers. So if there's no information on the surface of the nucleator, then we expect no information here in these EZs. The EZs are just generic EZs. On the other hand, if the nucleator has some information, so for example, this spot here and this spot here, there should be a minus here and a minus here, but instead there's either a 0 or a plus or what have you, which distinguishes this surface from other surfaces. Then you'd expect similar information to populate the EZ layers, as you can see here. So this would be, for example, the first EZ layer. And these so-called defects could then propagate over many layers, and therefore you get information. So you go from a generic EZ to an EZ with some differences that convey information. Now, another way of imparting the information is through electromagnetic energy. So you take the water, you put electromagnetic information in, and you get some resonant vibratory modes, which then will vibrate at their frequency, and also you can get information out. So you can put information in and get information out, the information, the intermediate, being stored in the EZ. So input electromagnetic energy can create information. And how exactly does this happen? Well, we don't know, but you can imagine, for example, that EM waves could impact certain oxygen atoms, depending on the frequency and such, but not others. So you could imagine that this oxygen atom and this one are impacted by the electromagnetic energy. And here's the critical part, is that each oxygen atom has not 2 plus or minus, or 1 and 0, but 5 different oxidation states, -2, -1, 0, +1, +2. If you calculate the information storage capacity, I don't remember offhand, but it was something like 10 to the 8th or so greater than if you have only 2 states, 1 and 0. It's a huge difference of information capacity. So if you think about it, in huge information storage capacity, it not only can explain or potentially explain memory inside of water, but also it's possible that the information stored in easy water could eventually replace conventional information storage, in essence, an extension of Moore's law, where you get smaller and smaller and smaller each, I think, 18 months or so to store the same amount of information. And so, and the question that's of interest here is, will easy water retain information following succussions and dilutions? And you'll hear from Vladimir Vyakov this afternoon, who's going to be talking about that and talking about retaining so-called nano aggregates as you do dilutions. And I think it's possible that when you do a succussion, you shake it really violently. And we know about EZ that if you do that to EZ, it breaks up the EZ into little tiny bits each time you do that. And it doesn't require more than just a bit of shaking, but when you do this, for sure, the EZ is broken up into tiny little bits. And I think it's possible that these tiny little bits can explain some of what Vladimir will be talking about, the retaining of nano aggregates which may contain information. And as you dilute, since you have so many of those, this information should be retained after successive dilutions. So this could be really important in understanding about homeopathic dilutions and why you're not just diluting out, but you're retaining information. Now, I know that a lot of homeopathic dilutions are not in water, but in ethanol. We studied to see if ethanol also produces exclusion zones, and the answer is yes. So this is the ethanol volume fraction going from 0 all the way up to close to 100%. And you can see that in this particular experiment, the pure water gives you a roughly 80 micrometer exclusion zone. And as you change the fraction to get toward 100% ethanol, you still get an exclusion zone. It's less, it's about half the size with pure water. But the point is that if you use ethanol, you get an exclusion zone pretty much the same as water. And we haven't studied the structure in terms of ethanol, but it looks as though there's a kind of similarity between the two. So any argument that is based on the idea that homeopathy is often in ethanol rather than in water, I think this would tend to dispel. So the answer to question 2, and I'm sorry this has been a long segment, that question, is the EZ physically distinct from bulk? The answer is yes. Seems to be a layered honeycomb structure, and it has information storage capacity. Now, question 3, can crystalline water explain counterintuitive anomalies like the ones we started with? What behavior do we expect from a crystal? Well, one of them is that crystalline elements stick together. And if you think, for example, of gelatin dessert, it's 95% water. Right? Do you ever think of when you put the powder in the water, 95% water, and you get a gel, how come the water doesn't dribble out? It's something not all of us have a tendency to think about, but you think about it. So some scientists, some chemists will say, oh yeah, the answer is really simple. It's the osmotic pressure. So if you have some solids, the solids tend to pull water toward the surface. But if you think about quantitatively, I've handled some gels in Japan that are not 95% water, but 99.95% percent water. It's essentially all water and a few little strands of polymer, and the water doesn't leak out. And so how is it possible to explain this by osmotic force? And I think you can explain it osmotically, but there's another explanation, and that is if you look at a gel, the structure looks something like this. It's got either a polymer or a protein matrix, and inside of that matrix are these gaping holes. And what are the holes filled with? Well, in a hydrogel, just like ordinary gelatin dessert, the space is filled with water. But what kind of water? Well, we know that if these surfaces are hydrophilic, which they are in the gel, that they're going to take the water and convert the water into EZ. So you get layers, 1 layer, 2 layers, 3 layers. So the water that's inside here is not ordinary H2O, it's EZ water. And by the way, if you stick an electrode in, you get a negative electrical potential inside the gel, just like in the cell. And we know that EZ water is gel-like. And so if you ever ask yourself, how do you get this weird consistency that you see in, for example, raw egg white? It's sort of, I don't know, first time my mother showed it to me, I thought it was kind of disgusting. But anyway, I think the answer is nothing to do specifically with the matrix, but has to do with the water. The water, which is a major part of it, has gel-like, EZ has gel-like properties, and that's why the gel has the feel that it does. Now, another question is what about the surface of the water? Some of you know you floated a pin or a paper clip on the top of the water. You place it carefully and it floats, but it shouldn't float because it's more dense than water, but it does float. If you put it beneath the surface, it sinks, but on the surface, it can float. And so you may think, oh, it has something to do with surface tension. But surface tension affects one molecular layer on the top. And the question is can you really explain this phenomenon like what you see here, which is an old Hungarian coin with one molecular layer? And so we investigated to see what happens at the surface of water. And we found that the EZ water appears to grow at the air-water interface. And the next slide will show you what's going on. So what you see here is a chamber, and this chamber is built of 2 pieces of glass brought together and sealed around the edges to make a chamber. And then you pour into it water and microspheres, just the standard stuff that I've been showing you, and it's cloudy because the microspheres scatter light. So you have air here, and then you have the meniscus sitting here. And what we found is that you get a clear zone that builds up in a matter of 15 or 20 minutes or so, and the clear zone is essentially microsphere free, and so it looks similar to the EZ. We stuck an electrode in, and we confirmed that it has a negative potential, just like the EZ. And then in the next slide, I'm going to show you that the behavior of this negatively charged, looks like EZ, it behaves like a thick gel-like band, not like ordinary water. And so it's shown here. Here are the microspheres in the water, and here's the clear zone. And what we're going to do is we're going to push down on this glass rod. It's going to touch the surface, and it's going to mechanically perturb the surface. And what you see is that the thickness of this zone doesn't change noticeably at all. And so it's shown here. So you're perturbing the surface mechanically, move it back and forth, and this zone behaves like a coherent gel-like zone rather than ordinary water. So we've concluded that you have many structural layers that create the high surface tension, EZ-like layers, not just a single molecular layer. And this is essentially EZ that occurs at the surface. And that may help explain this slide, which some of you have seen before. This is a lizard in Central America, our neighbor, and it sits on a branch of a tree. And when it gets the urge, who knows what the urge is exactly, it jumps from there and it walks on the water. And because it walks on water, it's known as the Jesus Christ lizard. And you can see it here. And the question is, can you explain this by one molecular layer? Or I think better, you can explain it by a thick layer with many millions of layers. And the same thing, remember the water dropping on the surface? Remember, it doesn't coalesce instantly. So remember, the water that's sitting down here is exposed to the air. And the air-water interface, as I showed you, has a thick EZ at the surface. And pretty much the same occurs here because this droplet of water is interfacing with the air. The pendant droplet is sitting there for a while, and so you get an EZ that forms around the droplet and also down here. So when the droplet falls on the water, it's not water meeting water, it's EZ meeting EZ, you see. And it takes some time for this to be breached, and I think this is the reason why you see what you see, which is characteristically approximately 5 stages, boop, 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 a little kind of dance that it does. So another point is that crystals can be stiff. Some of you have crystals of a ring or some jewelry, and you know that these crystals are stiff. Some are less stiff, but some of you have played around with salt crystals or sugar crystals, and they basically stiffen. And so if you go back to this structure and think about how come this can persist. I mean, this has huge stiffness. You can calculate the stiffness based on the length, the amount of droop, the thickness of this, and it's very stiff. You can almost walk across it. And how do you explain this? Ordinary water doesn't behave like this. You've never seen water behave, but if you have a crystalline kind of water, under the right circumstances, it can become stiff. So the answer to question 3 is yes. Liquid crystalline water explains many anomalies. Now, the critical question is what charges this water battery? So we have EZ negative, and you have the positive zone right next to it. So it's like a battery. Every battery needs charging. Your cell phone battery will run down, and you have to charge it again. So where does the energy come from? And I must admit that this was a seriously confounding problem for us. We couldn't figure out the answer to that question. You know, obviously you can't plug the chamber into the mains and get some voltage there. So where on earth does it come from? And it took us a few years to figure out the answer. But I got to tell you that I asked the question when I'm teaching the students. I usually give a kind of introductory lecture to this stuff. And I asked the question the same as I would ask to you, where does the energy come from? And one of the students timidly raises his hand and comes up with the right answer. And we quickly got this guy to work in the laboratory. It took us years, but and the answer is that it comes from light, incident radiant energy. How did we find out? Well, it was one of the students working in the lab. We have a lot of undergraduates who like to volunteer and work in the laboratory. And the student will typically be assigned to a project. We discuss one project, and they can give them a choice, and they choose something. But they often deviate. And you know, I pretend that I don't like when they deviate, but actually I do like when they deviate because sometimes they find something interesting. So this student is sitting on the bench, and he's got a chamber here, and there was a gooseneck lamp. So he shines the light on the chamber, and here's what he found. This is actually real data. This is not real, but he shined the lamp. And so this is a piece of Naphion, and here's the exclusion zone, and here are the microspheres. And in the region that was illuminated, he could see the EZ growing like that. And if you pulled the lamp away, it would collapse back down to a straight EZ running across, time constant of 15 minutes or 20 minutes or so. So it didn't take a rocket scientist to figure out that, you know, if you shine the light and it gets bigger, that maybe light is responsible for growing this exclusion zone, or I guess as physicists would say, electromagnetic energy, which encompasses visible light plus other wavelengths. Well, naturally, we were excited by this observation, and we did real experiments. And I show it in a small panel because I don't want to spend time on it, but basically this is wavelength, and this is EZ expansion, and there's a peak over here. And the peak is actually much higher, but for technical reasons, it's here. And this peak is at a wavelength of 3,000 nanometers or 3 micrometers. That's in the infrared, not visible. So we went from UV all the way to visible and infrared. And infrared is by far the most effective. And so, yeah, here. So where does infrared come from? You know, many people are not aware. We think of infrared, we turn on the toaster, you know, and it glows, the coils glow bright orange, and we think we're getting heat and we're getting infrared energy from that. But actually, infrared is far more common. If we were to turn off the projector here, it's pretty dark. We wouldn't be able to see anything. However, if I whip out my camera from my smartphone, which I don't have any, but we would get nothing. But if I took an infrared camera instead of a smartphone camera, the sensor is sensitive to infrared. I get a beautiful image of all of you, even though it's dark in the room. You know, I'd be able to see your tie and your watch and the seat and my wife's necklace. And yeah, no problem at all, because everything is generating infrared energy. It's all over. It's actually, you might say it's free energy, literally free, because we don't have to pay a nickel for it. It's absolutely free. So it's there all the time. And since it's there all the time, it must be that EZ is prominent, is present all the time. And so you have a situation that looks like this with a hydrophilic material next to water, and a certain size EZ occurs because there's ambient infrared energy that builds this up. And if you were to supply more energy, the EZ would build. And if you got rid of that energy, it would come back down. And in fact, if you block the energy, I don't want to take the time to show, it gets smaller. So the answer to question 4 about energy is that the EZ buildup is powered by light, photonic energy, and that orders the water and charges the water battery. So the situation is kind of like Hawaii. You just relax in the sun and you get a lot of energy. So I'm suggesting to you that this water is actually receiving energy from the environment. And so if it's receiving energy from the environment, you'd expect that you ought to be able to capture the energy in some way. But I think not one of you has ever seen a glass of water doing work, right? And I'm going to show you that a glass of water can do work. You can actually harvest the energy that you get from this kind of battery. And the situation is like this. So we took a piece of a tube made of naphthalene, just like a straw. We found that we could get it, and since we got it, we thought, hey, let's play with it. And one of the undergraduates was playing with it, and I asked the undergraduates to see if the tube, just like the flat sheet of naphthalene, could grow an exclusion zone right next to it, outside and perhaps inside. And the student was playing with it, and one day the student came running into my office, and I had someone sitting in the office. I don't remember who it was because I think it was an important person. But the student came barging in. He didn't care that the person was important or not important. And he said, do you mind? I found something that you might find interesting. And I actually didn't mind. And he came and he said, you know, I found that the water keeps running through the tube and it never stops. And he thought it was important enough to come run and tell me. And I thought it was even more important because, you know, usually when you have a tube, in order to get something to flow, you need to apply pressure to one end or a pressure gradient across it. There's no pressure gradient here because the pressure at this end and this end are equal because this is lying horizontally. So there's got to be some other energy that's responsible for pushing it. It's like your large blood vessels. You've got the heart that builds pressure and pushes the blood through. And the same situation here, which meant if the student's experiment was correct, it meant that there's got to be some energy that's coming into the system. And we had already identified that the environment, the energy, infrared energy from the environment was coming and getting transduced by the water. And therefore, that looked like the proper supply of energy. So the experiment looks like this. You take a tube, and you fill the inside of the tube with water, make sure that there are no air bubbles, you see there, and put it in water and microspheres, and then take it to the microscope, and we use green light just to restrict the amount of illumination, and you get something that looks like this. You see, and it keeps going. We've had this going for as much as a day and a half without stopping. And then we thought, maybe this is something peculiar for Naphion, maybe. And so we decided to get tubes made of other materials. And what other materials? It's hard to find narrow tubes made of hydrophilic materials, at least we could. And so we created our own. And the way we did it was we formed gels, and we put a wire in the gel. And as the gel formed, we pull out the wire, and you're left with a chunk of gel that has a tunnel that runs through it. And you take this chunk with tunnel, and you put it into water plus microspheres. And we wanted to see whether we got the same result. So this is a polyacrylic acid gel here. And here's the tunnel, runs from here to here. And you can see that next to the polyacrylic acid gel is an EZ and an EZ here. And all the microspheres get squeezed to the middle. And if you look at the video, it looks like it flows the same way as the Naphion. We've now tried no fewer than a half dozen different gels, and we get a similar result, except that the velocities tend to be different depending on the particular gel. Now, someone said to us, well, gee, you know, if you think this is all powered by light, electromagnetic energy, why don't you just crank up the light and see if it goes faster? So we did that experiment, and we published a paper 2 years ago showing that we could get up to 5 times the speed increase just by turning up the light. So this is definitely driven by light. And so you have a situation that's like this. You have a hollow tube, and work is done because the water has certain viscosity, and to drive it through, because of frictional losses, you must need energy to drive it. And so energy is required in order to achieve this. And so where does the energy come from? Well, we already showed that the water is absorbing energy from the environment. So that basically, the water is transducing the light into EZ, which is actually responsible for driving it. So this water is actually a transducer that takes light energy and converts the light energy to mechanical energy if you have the right circumstances. That sounds weird. Please. Good question. The short answer is we're not sure. And the long answer is that, or longer answer, of course we've thought about that issue, and that in order to go one way versus the other way, you need some asymmetry. And how do you create the asymmetry? Well, first of all, you don't always create it. 10% or so of instances we get no flow. And we assume that we get no flow because the symmetry is too great. However, in a typical experiment where you've got a light on one side or a window on one side, you have more energy coming in on one side than the other side. And we think that should be sufficient to create enough asymmetry to make it go one way or the other. Okay, thank you. Good question. So it's kind of weird, but just think for a moment about what's going on with the plant that sits on your windowsill. It's receiving light. It gets its energy from light. So light builds chemical potential, and the chemical potential drives everything, essentially, from metabolism to growth, bending, you name it. And the results are suggesting that the same thing occurs in the water. And it's no surprise because the plant is mostly water, you see. So we think something similar is going on here, and that gives you an end result of an equation that looks like a familiar equation to some people, an iconic equation. And I know that the units don't match, so it's wrong, but I think you get the idea that this contains energy. It's not at equilibrium with the environment. It's got potential energy that can do work. Okay, so why is this all important? I think it could be foundational for any or all science involving water, molecules, and light, and also foundational for health. After all, we're mostly water, and we're also mostly EZ water. And because our water is almost always next to some interface, some hydrophilic interface. So if you want to know something about the body water, about biology, you need to know something about the water and biology. So the first question is, does human biology use radiant energy? You know, we're receiving infrared all the time. And the question is, do we just re-radiate it out, or do we actually make use of it? So if you're Mother Nature, and you've done pretty well with plants, with green plants and some bacteria, you use the light to get the energy, and you were creating animals, what would you do? Would you just discard it, or would you save it and perhaps use it as a backup? And I'm suggesting to you that maybe we actually might use the light energy that we get in the same way, and not exactly the same, but as plants and some bacteria do in photosynthetic mechanism. And so the first thing that comes to light is about the cardiovascular system, which is, much of which capillaries are right near the surface. And I got to admit a kind of personal arrogance, because I did my PhD doing computer simulation of the cardiovascular system, pressures and flows, and I thought for sure we had all the right answers, until I visited Moscow. Where is Vladimir here? Yeah, and Vladimir introduced me to his colleague from the laboratory next door, and the guy didn't speak English, but Vladimir is really good at translating, and so he said, there's a real problem in the cardiovascular system. And I said, oh yeah, really? Tell me. I know something about the cardiovascular system. And he went on to say that there's a big problem. Okay, so what's the big problem? The big problem is that you have capillaries, and the diameter of the capillary is, in healthy young adults, not me, but down to 3 or 4 micrometers. But the red blood cells that need to go through are 6 or 7 micrometers. You know, so it's like taking a football through a narrow pipe. You got to push it through, and in order to get it through, the red blood cells have to bend. And in order to do this, you need energy pushing this squinched red blood cell through. So he said he calculated how much energy would be required, or how much pressure the heart would need to develop if the heart were fully responsible for pushing it through. And he calculated that it would need to generate a pressure of something like 1 million times the pressure that we generated. That would be really high blood pressure. So in most of us, that obviously doesn't happen, you see. So there's got to be some more energy. And he was speculating about various kinds of energy from bubbles and other things. And I'm kind of thinking that, you know, it's pretty interesting, because we know we take a tube, we put it in water, and you get what looks like spontaneous flow. And so I'm wondering, is it possible that the same mechanism might operate in your and my cardiovascular system? And so that's what we look toward. Now this just gives an example. These are muscle striations, and you can see blood vessels that run through. Now the red blood cells are supposed to look like this, but as they pass through, you can see they're squinched down to the kind of shape. In order to get through, you've got to do that, and that's what requires energy. And so you can look at a video and see how this happens. And the guy over in the center is having real trouble getting through, and you know, it's difficult to push it through. And so we're wondering, since radiant energy, light, drives flow through the tube, might radiant energy help drive blood flow in our vessels? And so we had a piece of evidence from an Israeli group that was doing an experiment in mice, and they were examining blood flow in capillaries using optical coherence tomography, an advanced optical technique. And after they did their experiment, they properly sacrificed the mouse by clamping the aorta, and the mouse complied by dying within 10 seconds, but they found that the flow didn't stop. This was a surprise. It was greatly reduced in flow velocity, but it didn't stop, and it went on for more than 1 hour. And they did the experiment again, and they got the same result. They repeated it 10 times, and they got a similar result. And we've since found out that there are other groups who reported scattered over the literature over many years, who reported the same thing, that when the heart stops beating, the flow doesn't stop. So we confirmed it in our experiments, and we wanted to test to see whether the same mechanism that we found in the chamber might be applying in the heart. And the signature of this phenomenon is that if you apply infrared, it gets faster. So we did the experiment. We took a chick embryo. You can see the eggshell here, and the top of the eggshell is removed, and you can see the embryo here. This is 3 days old. This is the beginning cardiovascular system before much has developed. And this is done by a student in my laboratory. And this is time, and this is flow. So you stop the heart, and he does that by applying potassium chloride. And after the heart is stopped, the velocity goes down, but it's not zero. It maintains itself for some period. And then we wanted to see if we turn on the infrared light, does it grow faster? And you can see that it does. It increases by 3 times or so, and then you turn it off, and it goes back to the control. So we think that this mechanism is actually operating in our cardiovascular system. And the infrared energy that's driving it could come from outside, but also all the metabolic reactions that are occurring inside of our body are generating heat and therefore infrared. So the heat that our body generates is not necessarily going to waste. It's going possibly to power blood flow in our capillaries, to augment the power that's generated by the heart. So yes, now does this radiant energy apply only to the cardiovascular system or other cells in our body? And I think the answer is yes. We use this energy in all the cells of our body, and let me explain how I think this is occurring, because it's very important. We think that the only energy we get is from food, but I'm suggesting to you that we may get energy from light in a way similar to plants. And so if you think about the cell, here's a membrane, and here are some macromolecules. And the surfaces of these molecules are typically hydrophilic over most of their surface, and therefore they generate negatively charged easy water. The cell is very crowded. Even this underestimates the crowdedness of the cell. So basically it's filled with easy water. And as I mentioned, if you stick an electrode in, you measure negative electrical potential. There are conventional explanations for it, but I think the correct explanation is that the water is negatively charged, and that's why the cell is negatively charged. Anyway, negative charges repel one another. They want to get away from each other, and that's potential energy, you see. And the potential energy is basically driving a protein folding. And protein folding, the movement of protein from this state to this state, is how the cell does its work. Whatever cell, whether it's a cell that contracts or a cell that secretes or transmits information, it's always protein folding from state 1 to state 2. And it looks something like this. So you have a protein, and the protein is surrounded by easy water, and it's the melting of the easy water that allows the protein to fold. And as the protein extends itself back to the original state, the easy water grows again, and the resting cell is filled with easy water. Now, if you don't have enough, or at the extreme, if the easy water is absent, then the protein is in an unfamiliar circumstance, and it can't do what it's supposed to do. And so you have a situation that sort of looks like this, and it's inept. And basically what you need to do to bring this back to normal function is to supply easy water. The cell must have its proper complement of easy water. You might say it's properly hydrated, because this is hydration. It needs to be hydrated. And so the major point is that this potential energy from easy is driving the work of the cell. So the sequence looks like this. The light is building easy. Easy has negative charge, and this contains potential energy, and the energy is used to drive the work or folding of the proteins. And if you connect the dots from here to here, light is being used for the work of the cell. And so the question, where do we get our energy? Well, of course we get our energy from food, some of us too much. But we also get energy from light. And the light is basically absorbed by the water and builds easy water, which gives us energy. So it's not just the food, it's also the light that gives us energy. Should this matter to you? Well, yeah, water matters and light matters. And so if you think about health and maintaining health, one approach is asking what builds easy water in your cells. And so one thing that helps build is to drink water, because water is the raw material for building easy water. So if you don't drink enough, you don't get enough, and your function goes down. So you need to make sure you drink enough. Another is green juicing. I know some of you know about that. You take the plant, a fresh plant, and you squeeze the juice out of it. So what is this juice that you're squeezing and drinking? It's the juice it's the inside of the plant cells. And the inside of the plant cells, like the inside of your cells, contains easy water. We tested this and checked it. So you're basically drinking easy water. And what could be better than replenishing the original with easy water from somewhere else? So that's why many practitioners report that one quick way of regaining health is by drinking juice from plants, from raw plants. There are also a lot of material back from Ayurvedic times and beyond, substances that have been known for a long time to promote health, and promote health over the entire body, not just in one organ or organism. And we tested these. We tested turmeric, coconut water, coconut oil, ghee, half dozen of these, and we found that all of them build easy water. If you take a poison, on the other hand, like glyphosate, which is in Roundup, it does nothing but diminish easy water, you see. So it may be that these agents that have been known for so long to promote health by actually increasing the hydration of the cell. Sunshine, if you're from Seattle, you crave it. And we walk outside, we see the sunshine, we feel good, we think it's psychological, but the light builds easy water, you see, and this could be the reason. And then it comes to the sauna. Infrared light, we know it feels good. You come out of the sauna, you walk in a wreck with muscle aches and pains and a headache and feeling tired. You come out after and you feel like €1,000,000 or pounds or dollars. And the question is, well, how come? Well, the sauna is generating infrared energy. Your body is absorbing infrared energy. The infrared energy builds easy water. And if you build easy water back to the proper amounts, then your organs are functioning properly and you feel good. And finally, there are more, but earthing. What about earthing? Connecting yourself either to the earth, either by walking barefoot on the beach or using other ways to connect yourself to the earth, which we don't ordinarily do. Well, the earth is filled with negative charge. I didn't know this until recently, but you can find it in Feynman's lectures and such, that the earth has a net negative charge. And so by connecting yourself to this net negative charge, you're allowing the negative charge to seep into your body. And what negative charge does is build easy water, which has negative charge. So this is another possible way. So I just listed a half dozen simple ways that are actually known to promote health. And I think there's a common mechanism for all of these. And the common mechanism is the buildup of easy water, which you need for function. So I'm not suggesting that we photosynthesize. But I'm suggesting that we use light. And those of you who know about photosynthesis, step one in the 20 or so steps of photosynthesis, step one is the light splits water into negative and positive components. That's exactly what we found here. So what we found could be a kind of generic photosynthesis. Now, finally, practical applications. If this stuff is not practical, maybe what's practical? So one of them is getting energy from sunlight and water. And this situation, by now, you're familiar with it. And so we put one electrode here, and in theory, we ought to get electrical energy out of it. And I showed you a record showing current flow between the two. And one of my students, Kurt Kung, has actually implemented this, and this is his setup. So here you have not one chamber with Nafion, but I think about 100 or so with Nafion. This is a switch, and this is a magnifier, and there's an LED that sits under, and when you flip the switch, you can see the light goes on. So we're getting electrical energy out of water, It's water and light. It's transducing energy from the environment to give you electrical energy. So this is one of the outcomes that we're pursuing. We have a company, no surprise, it's called Fourth Phase Incorporated. That was not my idea. And we're pursuing this and also pursuing getting drinking water from contaminated water. And you may have surmised that there's a kind of simple way to do this. So basically the heart of this is a tube of Nafion. And flow goes into the tube, and this flow may contain water with all kinds of junk in it, contaminants, and as representative junk, we use microspheres, the little particles that I showed you. And remember, there's an exclusion zone here, and an exclusion zone here, pushes all the particles to the center, and what we have is we built this so-called differential extractor, which is a fancy name for two concentric tubes. And the first one collects the water at the center and dumps it, and the other one collects the water from the periphery, which is EZ water, and you can see that there are fewer microspheres here than here, and we've been able to achieve in a single pass, we've been able to achieve a separation of 200 to 1. And of course, the amount of water that this produces is maybe just enough to satisfy the thirst of a flea, but we've improved it, and now we can collect a lot more water. And the dream that we have is being able to put salt water in and get the salt dumped here and the pure water coming out this way. We have some experiments that the company is doing which suggest that there is the possibility, but the results so far have been somewhat inconsistent, and so we're working on this right now. Anyway, those are two potential applications of the science that we've done. So I will conclude by saying the main point in general is that we think that water has not 3 phases, but 4 phases. The 3 conventional ones are ice and water and vapor, but we found evidence for an EZ phase. And I put the EZ phase in between the ice and the water. If you remember, the structure of this, or at least the deduced structure, is not so different from the structure of ice. And we have now experimental evidence that if you want to freeze the water, you start reducing the temperature, and as you reduce the temperature, the EZ grows, and then the EZ is actually converted to ice. And if you melt the ice, again, evidence that the ice first turns into EZ water, which is why many people like to drink freshly melted ice, which then converts into ordinary water. So this is a necessary intermediate between here and here. And so what I presented to you, I think, has broad implications. To summarize, the central point is that this water is receiving radiant energy from the environment all the time. And there are biological applications. I gave you one example about blood flow in the cardiovascular system, and yours and mine, which the evidence suggests is partially driven by radiant energy from outside. And in terms of chemistry, if you read the chemistry books, you can look at the interpretations of all the reactions, but in no interpretation is the effect of light or EZ water or separation of charge discussed at all. So it might be that some, maybe many, reactions will need to be reconsidered interpretation. In terms of weather, next book coming soon, we think of weather in terms of pressure and temperature. And I will like to demonstrate to you, and this is a next book, a different talk, that charge in the atmosphere and the clouds is far more important than any of these variables. And if you really want to understand weather, you need to start with that kind of premise. And EZ water is stored in clouds, just like you saw a beaker containing clear water and clouds of EZ water at the center. In terms of health, water is critically important, and since most of the water inside the body is EZ water, in order to understand the basis of health, you really need to understand the dynamics of EZ water. If you want to preserve food by dehydrating or freezing, it's helpful to know something about those processes. And then there are so-called practical applications like filtration of water and perhaps desalination using this technique and getting electricity. In terms of homeopathy, I think the most important aspect of what I've shown you is that the EZ water, being a semi-solid, regularly ordered structure, has the capacity of memory. I'm not sure it has the capacity for thinking, but it has the capacity for... And finally, before my final slide, I just want to mention something that is an aside. This has nothing to do with water research, but we have the Institute for Venture Science, something that we have been building, and the Institute funds promising ideas across all of science, nothing to do necessarily with water, that may bring scientific revolutions, of which there have been very few lately. We hope to change that. We're in the process of reviewing a dozen proposals, and there are a few people in the audience who have been either submitters of proposals or reviewers of proposals. And of course, we're ever looking for donors who would like to do something for humanity by supporting this kind of operation. We're very excited about it. Okay, and the last slide is, if you want to find out more about this, please have a look at the book. It's got pretty pictures in it, and it's written for lay people. So I'd like to stop here, and thank you. Thank you.
Thank you.